It's 12. If it's possible, I can start. I think you can hear me all, no problem.
Yes.
It's great. First of all, I want to welcome you all to our fifth ODAKtr fifth seminar by Dr. Inmaculada Polo from CIEMAT-PSA, Spain. Today, the seminar will be moderated by Assistant Professor Dr. Zohre Kurt from Middle East Technical University Environmental Engineering Department. Zohre Kurt is one of the main researchers in joint research lines, two joint research lines in our Solar Twins project. And now we are making the ODAKtr seminar series. ODAKtr is a national CST concentrated solar thermal initiative led by Metü Günam, Center for Solar Research and Applications, with the objectives of supporting Turkey's energy transition through the development and commercialization of CST technologies, catalyze the domestic CST economic activity, and strengthen Turkey's CST research and innovation capacities. I would like to give the floor to Zohre Hocam for the introduction. Thanks for participation.
Thank you very much, Yelda, and thank you, Inmaculada, for accepting our offer to give your seminar. Dr. Maria Inmaculada Polo Lopez is going to talk about a very interesting topic today. She's going to describe antibiotic resistant bacteria, its occurrence, removal from the urban wastewaters. It's a very important topic because, as you may all know, antibiotic resistant bacteria can cause lots of problems, especially when it is present in the pathogens. So being able to remove such bacteria is very important, especially for the for everybody's health, and for us who are very interested in urban wastewaters and keeping the safety. Dr. Maria Inmaculada Polo Lopez is a senior researcher in solar treatment of water in the Plataforma Solar de Almeria in CIEMAT, Spain. She has gotten her bachelor degree in biology. However, she moved to chemical engineering department by doing her master's and PhD in University of Almeria, and she stayed working on her photocatalytic disinfection of water and wastewater for drinking and reuse purposes, which is again quite important because of the scarcity of water and the importance of water that is globally understood recently, as you might know. She's author and co-author of 72 publications, 2 books, and 12 book chapters. She has been in many research projects. She has also supervised and co-supervised PhD students, and she continues her work again in Almeria, CIEMAT, Spain. So it's a pleasure of us to listen to you, Inmaculada, and your research, and understand how we actually could use solar energy to be able to treat antibiotic-resistant gene and antibiotic-resistant bacteria from wastewaters.
Thank you.
The floor is on you.
Thank you, Zohre, and thank you, Jella, for the invitation and for the kind introduction. I'm going to share my screen to show the presentation.
And please, if you have any questions, you can use the chat, and after her talk, we're gonna answer the questions.
Can you see? Can you see the presentation?
Yes, we can.
Okay. So, thank you for the introduction. And just before to start with the real contents of the presentation, I would like to give you just an overview of the research center called Plataforma Solar de Almeria, where I belong to and I'm working. Plataforma Solar de Almeria is an European largest scientific installation and it is being the largest and most complete research and development center in the world devoted to solar thermal concentration system. And PSA is also a singular science and technology infrastructure in Spain. The main goal of PSA is the research and development in potential industrial application of concentrated solar thermal energy, but also in solar photochemistry, where two groups are actively working in this last topic, which are the solar treatment of water, where I'm working, and also the solar desalination group. Plataforma Solar de Almeria is located in the southeast of Spain, and it is distributed in around 100 hectares in the Tabernas Desert in Almeria. Here you can see an aerial view of the PSA with the different installation, the big installation devoted to high solar thermal concentration system, and in particular the solar treatment of water is located in this area, where we have the laboratory and also the reactors installed outside. Here you can see a view of the facilities of the reactors. Most of them are the compound parabolic collector reactor, and we have many different pilot plans for different research and different objectives that we are using along during we have developed all this installation during the last 20 years that we are working in the water treatment of water. The main research activities of the group are summarized here, and just to give you what we do, a part of the topic of today, is the following research activity lines. The first one is the assessment of solar photocatalytic and photochemical processes as tertiary treatment for wastewater, with two objectives: the removal of pollutants and also the removal of water pathogens. We investigate also the possibility of integrate solar advanced oxidation processes with other advanced treatment technologies such as nanofiltration, ultrafiltration, ozone, in ultraviolet C, for the remediation of industrial and also urban wastewater, and for the removal of pollutants and pathogens, always with the aim of improving the water treatment efficiency and also reducing the operating cost. We also assess the photocatalytic efficiency of new materials developed to be used under natural sunlight, and also to be used in the compound parabolic reactors at pilot scale. You can see here some views again of the solar reactors. We have also another line which is focusing on the solar photocatalytic generation of hydrogen and the visible lights with active materials at pilot reactors for testing. That this is in particular this picture. Also, using solar photocatalytic and photochemical processes for water disinfection is the other line of activity. For the purification of contaminated water sources with a high number of water pathogens. And finally, we also develop, test, and assess new concept of solar photoreactors at pilot and also at demo scale for both the water decontamination or disinfection and for different end purposes: water reclamation and reuse, and drinking water, and so on. So after this brief overview, let's go to the contents of the presentation, which is focused on to see an overview of the current situation about wastewater, and specifically the presence of the antibiotic resistant bacteria. We are going to see an overview of the antibiotics, because we cannot speak about antibiotic resistant bacteria without to know the antibiotics, the development, the spreading, the occurrence in urban wastewater, and how the different conventional tertiary treatment typically already installed in wastewater treatment plants and other solar treatment can remove these particular targets from the water to make this more available and more safe for reusing. And finally, we will see some concluding remarks. Well, about the antibiotics, as most of you already know, antibiotics are chemical substances capable of paralyzing the development of certain pathogenic microorganisms due to its bacteriostatic action, or causing death due to its bactericidal action, that and they are produced by another living microorganisms or are manufactured by synthesis. The first discovery of antibiotics was by Alexander Fleming, which discovered for the first time the penicillin. This antibiotic means the revolutionate area in the medicine, because from this first discovery, a high amount of other antibiotics were discovered and also synthesized. So now we account with a big number of antibiotics to kill other microorganisms, and in medicine seems one of the most important advances that we have in medicine for treating infections. The antibiotics has different modes of action. Depending on the mode of action, the antibiotics are classified. We can see in this slide and in this figure, very brief, the different mode of action of the antibiotics, that we can focus on the inhibition, preventing the multiplication of the bacteria, which are, for example, the inhibitors of synthesis of folato, and some disruptors of the synthesis of the cell membrane synthesis. Other antibiotics can also generate the inhibition of the synthesis of proteins, because they are affecting different parts of the architecture and the machine of generation the proteins, such as you can see they are affecting different areas of the ribosomes where the proteins are generated. There are other types of antibiotics that affect the alteration of the DNA structure, such as the inhibitors of the DNA-dependent RNA polymerase and other affecting DNA generase inhibitors, and also other antibiotics that inhibits the cell wall synthesis. All of these actions are the typical mechanisms where the antibiotics can inactivate the microorganisms. In this table, you see again the different antimicrobials depending on the mechanisms of action, such as the inhibition of bacteria cell wall synthesis, the alteration of the membrane, the inhibitors of the protein synthesis, etc. You see how amount of different type of antimicrobials we have for treating infection. And in red are highlight the most used antibiotics for treating common infection, such as the amoxicillin, doxycycline, cefalexin, ciprofloxacin, clindamycin, metronidazole, azithromycin, sulfamethoxazole, and trimethoprim, amoxicillin and clavulanic acid, and levofloxacin. These 10 antibiotics are the most used, as I said before. And the future perspective regarding the use of antibiotics is that the use and the consume of these antibiotics will still increase in the next year. This is because two main reasons. The first one is the increasing of the global population. It's clear if there is more population, more infection will appear, and more antibiotics we will need to treat the possible infection generated, and also the appearance of the resistance. What that means, the resistance? The antibiotic resistance is a global challenge, and it is defined like a phenomenon by which the microorganisms is no longer affected by an antimicrobial. at which it was previously sensitive, so that the usual medical treatment become ineffective and infection persists and can be transmitted to other people. This is a real problem, because if we have antibiotics to treat some disease and this cannot be worked, we need to look for another one. Sometimes the microorganisms has generated resistance to different type of antibiotics, so we cannot treat. And this finally generate infection that can be not treated, and people even are died due to this effect. Here you see some data about the estimation and the perspective in the presence of the antibacteria and the antibiotic resistant bacteria, and you can see like it is estimated that by 2015 around 10 million of people could be dead due to the presence of this type of bacteria. So it's a real problem, and medicine is very important. And actually, we have a real problem in the medicine. Even though in February of 2017, the World Health Organization published a list of priority pathogens. This is the list. In this table, you have the different microorganisms with their corresponding antibiotic resistance to most of the more effective antibiotics that we have right now. And also they was categorized by priority, because these microorganisms, the infection generated by them, are quite difficult to treat. So regarding the development of the antibiotic resistance, we cannot separate the discovering of the appearance of the introduction of new antibiotics with the appearance of the resistance. This is a graphic where you can see the time between the introduction of the antibiotic and the appearance of the corresponding resistance. You can see in the time how different antibiotics were introduced or discovered, and then the resistant period. You can see that in years ago, when the era of the antibiotics started, this time was longer than recently, because of course we have developed more capabilities at laboratories and the scientific community to detect this antibiotic resistance. And these differences is because once the antibiotic started to be used, they must to be work for the susceptible bacteria, and then after a time of contact and the action of the selective pressure, the bacteria could be resistant. Then this bacteria should be propagated in the environment, and finally it will be detected. Regarding the mechanisms of bacteria to antibiotics, in this slide you can see that the resistance try to avoid the capability of the antibiotics to do damage in the bacteria. So the main mechanisms are based on mutation on the receptors of the cell wall, the development of efflux pump to expulse the antibiotics from inside the bacteria, modify the permeability of the antibiotics at the membrane level, the inactivation of the capability of these antibiotics by new enzymes, and also the modification of the place of action inside the microorganisms. And so with these mechanisms, the bacteria can be resist the presence and the action of the antibiotics. Regarding the types of this resistance, we must to know that there is a natural resistance, because this is due to the architecture of the microorganisms. some microorganisms has natural resistance because its permeability, for example, to the compound or to the lack of the target, where the antimicrobial agent exerts its action. There is an example, for example, like mycobacteria that are resistant to beta-lactams antibiotics because they have a lack of the cell wall. So there is not the target where this type of antibiotics can generate the damage. Or some Enterobacteriaceae, gram-negative bacteria, to the antibiotic vancomycin because it has a very large size. And some gram-positive bacteria to resistant to polymyxin B. CIEMATs act as on cytoplasmatic membrane, and they are highly protected. But the most important resistance is the acquired. This is the most frequent type of resistance with the greatest clinical relevance. And it is found in those microorganisms that were initially sensitive to a certain antibiotic, and that through various mechanisms that we have explained, have acquired the ability to be resistant to the compound. And how this resistance are acquired? The initial resistance appear due to the mutation in the genetic material of the microorganisms. Here you see cells, the DNA, and also plasmids. Plasmids are small circular and double-strand DNA molecule, independent from the bacteria DNA. And they naturally occur in bacteria cells, and often they carry genes providing genetic advantage, among them the resistance to antibiotics. And this plasmid, when appear mutation and they transfer to other cells, this is the way of spreading the antibiotic resistance. Here you can see that there are two main different mechanisms to spreading the antibiotics resistance. One is the vertical transfer because of the multiplication. When we have in a sample in the environment a population where cells acquire the capability to resist the presence of the antibiotics, the non-resistant will die, and only the resistant will alive. This resistance will be developed by multiplication, and finally this will be the win strain, and from here this strain that has the capability of resist to the antibiotics can also transfer this resistance to another strain. That this ways the so-called horizontal transfer of genes by the transfer of the genetic mobile elements, typically the plasmid. There are three possibilities, like you can see here, but we are not going too much inside of these mechanisms because they are complex. But you must to know that there are different ways to transfer this antibiotic resistance. So this has been general information about the antibiotics and the generation of antibiotic resistant bacteria. And recently, during last year, the presence of this type of target has been started to detect in urban wastewater. And this is very important because the urban wastewater are using for another use, and is the presence of these antibiotic resistant bacteria are not removal from the wastewater, can be spread more and more and more. And we need to avoid that. Regarding the estimation for the next years, the prediction of the antimicrobial resistance is very worse in some parts of the world, as you see here, especially in those countries with highlighted in dark blue, where the prediction is detected like a high abundance, because they will have low or even absence of a proper wastewater treatment, like you see in the maps. But why? What are the reasons to appear the antibiotic resistant bacteria in wastewater? Because as you know, wastewater treatment plants are installation where the residual water that contains bacteria, genetic material, and also antibiotics are together for a certain time. During the line of treatment of the wastewater, although these targets are removable, not are completely inactivated, and they have a contact time and enough to favor the appearance of the antibiotic resistant bacteria and genes because of the positive selective pressure, and it is favored the transfer of the genes by both modes, the vertical and the horizontal transfer of the antibiotic resistance. So at the end, we have like a shaker where all these targets are together, and they are generated this antibiotic resistant bacteria. We have detected many different types of antibiotic resistant bacteria in the wastewater. Regarding the typical or natural occurring bacteria that are present in this type of water, and that they are susceptible to acquire the antibiotic resistance, such as the Escherichia coli, Enterococcus faecalis, Pseudomonas, Salmonella shigella, Klebsiella, and Acinetobacter. All of them included in the priority list that was published by the World Health Organization. In this figure, just I would like to show you how the abundance of E. coli and Enterococcus as an example, and also the presence of the corresponding antibiotic resistant to different antibiotics, such as the tetracycline and sulfonamide. You see, like the concentration in the between the influent and the effluent of the wastewater in a treatment plant is different. It is decreased, but even after to the end of the typical treatment line in the secondary treatment, the concentration of bacteria is still detected, not only the typical and the natural occurring bacteria, but also the resistant one. Regarding antibiotics, also there is information. There is a lot of information of different antibiotics that can be detected both in the influent and also in the effluent of a municipal wastewater treatment plant. In general, this is the most commonly antibiotics detected. In the influent, we can observe an average of concentration of higher than 500 nanograms per liter. While in the effluent, the concentration is lower, but it is still being detected, like in an average of 100 to 200 nanograms per liter in general of these antibiotics. Regarding the genes, because we cannot forget that the genes is the key point on this aspect. We can see here, depending on the type of antibiotics, we can see different genes that has been detected and that are used by the scientific community to detect the presence of these antibiotic resistant genes. In red are the most usually used genes for that detection, and also the concentration in depending on the different type of antibiotics. Here continues with other groups of antibiotics, like you see, all and in red are the highlighted. In this figure is summarized the more abundant and dominant antibiotic-resistant gene detected in influent and also in the effluent of wastewater treatment plants. There are mainly belongs to these four groups of antibiotics: the beta-lactamics, sulfonamides, tetracyclines, and also macrolides. And the most abundant antibiotic-resistant genes are those called BLA, SUL, TET, and also ERMB. If we see again, we observe a reduction on the presence of this of the antibiotic-resistant genes if we compare the effluent and the influent and the effluent, but in the effluent is still being detected. So as you can see, in general, the secondary effluent of a wastewater treatment plant is still containing antibiotic-resistant bacteria, antibiotics, and also the corresponding genes that if we don't remove from the water, they go directly to the environment, or if they are used for another activity, they can spread in the environment, and this is a problem because they can reach people and they can generate this very important disease. So to avoid that, it's important to apply an effective and efficient tertiary treatment. And from now, we are going to see the most conventional technologies for the removal of bacteria and the capability and efficiency of these conventional technologies, and then the solar upon oxidation processes also to remove specifically the targets of the antibiotics, antibiotic-resistant bacteria, and their corresponding genes. In general, the conventional technologies can be divided in the physical removal, which means that the bacteria are not inactivated, are just only removal, and we have treatment like coagulation and sedimentation, but also filtration, mainly activated carbon, but the most important is the membrane filtration. Regarding those conventional technologies that can kill the bacteria or inactivate it are the chlorination, ozone, ultraviolet C, and also advanced oxidation processes that this last group could be divided in photochemical and non-photochemical processes, like you see in the slide. We will see more. I will explain you more in the next slide. Okay, regarding the membrane filtration, as all of you know, it can provide an effective measure to reduce the risk resulting from the release of bacteria, but also genes into receiving water bodies and the spread of the antibiotic resistance in the environment. However, it has an important drawback that is related to the concentrated obtained during the membrane filtration that must be treated effectively to inactivate the bacteria and remove and inactivate or degrade the antibiotic resistant genes. There is a different type of membrane filtration depending on the pore size of the membrane, and this is divided in microfiltration, ultrafiltration, nanofiltration, and reverse osmosis from the highest pore size to the lower pore size, like you can see fine in this very well in this figure. Like depending on the pore size, the capability of removal of retain targets and molecules are increasing. Well, regarding the antibiotic resistant bacteria, I show you here a very recent study regarding the capability of the membrane filtration to remove especially bacteria, but also especially the antibiotic resistant genes in free DNA and also in plasmid. In particular, in this study, it was evaluated the antibiotic resistant gene for the antibiotics canamycin and ampicillin in ultra-pure water using different type of membranes. In conclusion, from this study, and it can be obtained from this type of technology, is that nanofiltration and reverse osmosis with molecular weight cutoff lower than 1 kilodalt were effective for removal of the free DNA, while membranes with molecular weight cutoff higher than 1 kilodalt permits higher removal of plasmid DNA, but incomplete removal of free DNA. So it's important to consider this range of molecular weight cutoff for the application of this treatment in the wastewater. Another process is of course, the use of chlorination. Chlorination have applied in wastewater, and also is a powerful herbicide. It can control flavor and odor of the water. Also, they can control the regrowth and control the organic chemical compounds. It has some important drawbacks, such as the generation of the cold disinfection byproducts, which are compounds that can be carcinogenic and mutagenic. So chlorination is not the best option to treat wastewater because it contains a high amount of organic matter. However, it is using, but it must be avoided. Normally, the application of chlorination is using chlorine gas or chlorine solution. That after the adding, they are generated hypochlorous acid, which is the efficient agent to inactivate the bacteria. And it's depend on different parameters, such as, of course, the amount of the hypochlorous acid generated, and also the pH. The range of pH where chlorination is more effective is at pH 6 and 7. There are also some studies in the literature regarding the capability of chlorination for removal of antibiotic resistant bacteria and genes. Here you have some results where different dose of free chlorine were investigated for the inactivation of bacteria and also for the inactivation of the corresponding antibiotic resistant genes. But in general, in all these studies from the literature, the main conclusion is the same, is that chlorination is highly efficient for the removal of bacteria, but has a very limited effect on the degradation of the antibiotic resistant gene. So although the bacteria are killed, the genes still being in the water. The problem is that could be spread again when they go to the receivers' bodies of this water. Okay, another treatment very well implemented and investigated and used for the treatment of wastewater is the ultraviolet C radiation. It consists of the application of radiation with wavelengths between 200 and 280 nanometers, and it's very effective for inactivation of bacteria because affect directly to the DNA molecules that absorb these wavelengths mainly at 254 nanometers. So that when DNA absorbs this radiation, generate alteration in the molecule of the DNA, typically so-called dimers of thymine, which destroying its functionality, and therefore finally it's causing the inhibition of the cell capability to growth and the multiplication, and the bacteria could inactivate it. Nevertheless, it has a very important drawback. This treatment is regarding the capability of the microorganisms to regrowth after the treatment if the enough dose of radiation has not generated the total inactivation of microorganisms. So here regarding, again, some studies that have evaluated the inactivation of bacteria and some antibiotic resistant genes, it can be concluded that the UVC effect is greater again in the inactivation of bacteria more than in the degradation of the corresponding genes. In general, dose of UVC radiation higher than 100 millijoules per centimeter square are effective for the antibiotic resistant genes removal, while those lower than this than the same dose, below 100 millijoules per centimeter square, have lower effectiveness for the antibiotic resistant gene removal, which directly generate an increasing of its presence in the effluents. Other problem in the waste on the application of this treatment to wastewater is that the complexity of the water matrix and the presence of different substances in the treated water, the UV dose requires to obtain a good inactivation of bacteria requires more than 10 millijoules per centimeter square. However, normally these doses are not applied because it's highly cost. This is another problem. And of course, as I mentioned before, the photoreactivation and the dark repair of bacteria associated with UVC dose that could not inactivate completely the bacteria and they can reactivate it. Because this treatment has not a residual effect in the water. Now, with the ozone is also another typical wastewater treatment. As you know, the ozone is formed when passing air or oxygen through an electric discharge. It is considered the most effective oxidant and disinfectant for using water treatment processes, but it is unstable and cannot be maintained in the water supply system. Therefore, when it is used as primary disinfectant, a secondary disinfectant is necessary to maintain the level of disinfection. It requires dose and very low contact time because it's very powerful. Is between 300 and 3,000 faster than chlorine for the inactivation of microorganisms. But sometimes, depending on the composition, chemical composition of the water, they can generate also potential toxic byproducts, like in the case of chlorination. It has to be generated in situ, and it must be used immediately, and they are expensive in operation and technically complex. But anyway, also this treatment has been used for the evaluation of the inactivation of antibiotic resistant bacteria, degradation of antibiotics, and antibiotic resistant genes, such as these graphics that I have extracted from a recent study in this topic. from this study, it was concluded that the low doses of ozone and hydraulic rotation time of around 10 minutes, it was sufficient and enough to degrade the antibiotics in the wastewater. Nevertheless, it was required high doses of ozone and higher time of contact time to inactivate the E. coli and the corresponding antibiotic resistant bacteria to trimethoprim or sulfamethoxazole, and also for the reduction of antibiotic resistant genes, which were not totally degraded, as you can see in this figure that represents the relative abundance of copy genes of different genes, as you can see here. It's one of these, and in yellow, the yellow bars represents the application of higher dose of ozone and higher contact time. You can see the reduction are not totally, but also you can see very clear how in most of the cases is the concentration of these genes after a certain time to finalize the application of the ozonation increase, which is one of the advantages of the application of many wastewater treatment. So in this study, it also suggests that it is required a post-treatment by other combination with other treatment, such as the biological filtration or even activated carbon absorption, to remove as much as possible the presence of these antibiotic resistant genes from the water. So apart from the conventional processes that we have seen, there is also the advanced oxidation processes, which are chemical treatment that permit the removal of organics, contaminants, sometimes inorganics, and also microorganisms in water and wastewater through the generation of the hydroxyl radicals. Hydroxyl radicals are reactive oxygen species. It's the higher effective for the removal and the removal of organic compounds and activation of microorganisms. All the advanced oxidation processes are based on the generation of this type of radicals to increase the effectiveness of the processes. There are two types of advanced oxidation processes: those that are non-photochemical, which means there is no need a source light. So that's the phenton, the combination of ozone with hydrogen peroxide, electrochemical oxidation, and also the use of ultrasound. But also we have the photochemical advanced oxidation processes, in which we have a source of light that can be UVC or also can be the solar light. And you see here in the list a combination of ultraviolet C with hydrogen peroxide, with ozone, other combination, and the last two processes that can be driven by using natural solar light. So let's focus on the application of solar advanced oxidation processes, because this is the processes that we are more focused on at the Plataforma de Almeria. Regarding the heterogeneous photocatalysis, just as general information, the most important catalyst is the titanium dioxide, but there is many scientific researchers working on the development of new materials for increasing the effectiveness of catalysts. And this is based on the use of semiconductors that, when in water and in presence of natural solar light with wavelength lower than 380 nanometers that can generate hydroxyl radicals on its surface that are used for the degradation of organic compounds or the inactivation of microorganisms. Other type of solar up and oxidation processes is the homogeneous photocatalysis, and the most important one is the photophenton. The photophenton is a process where we combine the use of an iron salt with hydrogen peroxide and with natural sunlight. When these three elements are together, we have a cycle where the iron is reduced and oxidized, generating the hydroxyl radicals. This process is very, very effective, but it has only the main drawbacks that is must be carried out at pH around 2.8 and 3, which for using at for wastewater, for example, and for other treatment of water, is a drawback because it requires a pre-treatment and then a post-treatment to modify the pH. And regarding the activity lines in the photophenton processes, currently the researchers are focused on certain new chelants of iron that permits to work with this treatment at neutral pH. Regarding the solar up and oxidation processes, not only is important the processes, it's important also the reactors that we use for enhance the capability of the processes. At the Plataforma, Solar Almeria, as I said before, we use the solar reactors called compound parabolic collectors. You have here a picture of one of them. We have many, as I showed you previously in previous slides. And these reactors consist mainly in this diagram, and you can see fine, a transparent tube made of borosilicated and an anodized aluminum mirror that act as a mirror where the solar radiation is reflected to the tube where the reaction are occurring. So we accelerate and promote the collection of the solar photons in the photoreactors, increasing, of course, the efficiency of the processes, generating more hydroxyl radicals. And this is just a diagram of a compound parabolic collector with a flow system. The water flow through the illuminated area and then go to a tank and go again for the illuminated surface of the reactor. This is the normal way to work when we apply solar oxidation processes. So now I'm going to show you some examples of degradation of antibiotics, antibiotic resistant bacteria and genes by the application of different solar processes and of the oxidation processes, other only phototypical processes, like the use of hydroxy peroxide with natural sunlight. And this is a diagram of work in this graphical abstract, where you can see the importance of use natural wastewater that typically here in Almeria we obtain from a wastewater treatment plant. And then the water is treated by the solar-driven oxidation processes. And then we apply different technologies and analytical methods for detection of the organic microcontaminants, the bacteria, and also the genes. And here we have the results regarding the inactivation of microorganisms this is an example from as I told you, from one study we developed with different processes, and I bring this study because we conducted all the evaluation of all these targets together, so we can see an overview of what happened with this type of treatment. We can see, for example, here the profiles of inactivation of fecal coliforms, and in comparison with the antibiotic-resistant fecal coliforms. In this part of the slide, we have the profiles of enterococci, natural occurring enterococci, and its corresponding degradation profiles of antibiotic-resistant enterococci. In general, I would like to highlight that there is not a big differences in the profiles of inactivation of the natural microorganisms regarding with the antibiotic-resistant microorganisms. You only see a lower initial concentration, which is normal, but the response and the susceptibility to the different treatment are similar in all cases. So the application of a treatment will not depend on the type of microorganism. There is no difference between the natural bacteria and the antibiotic-resistant genes. So from all the processes tested with natural solar lights and using CPC reactors, we see that the best treatments for the inactivation of this microbial population was the use of hydrogen peroxide alone, the photophantom at neutral pH, it was conducted at neutral pH, and also the use of titanium dioxide in combination with hydrogen peroxide. In this work, also we evaluate the microbial regrowth after 3 days of storage the water, and we observe that in some cases we see the growth of microorganisms, but in general we could avoid the regrowth of bacteria, especially on those processes where we have hydrogen peroxide as a residual agent concentration in the water after the treatment. Regarding the removal of pharmaceuticals, we evaluate in this work the degradation of three organic compounds: carbamazepine, sulfamethoxazole, and diclofenac. You see here the degradation profile by the different solar processes, and of course we observe different behavior because one compounds are degraded more easily than other one, like diclofenac, which is the lower resistant organic compound to be degraded. But from all these results, we observed that the best treatment was reached with the use of the semiconductor titanium dioxide, P25, in combination with hydrogen peroxide. And regarding the antibiotic-resistant genes that it was evaluated also in this study, we detected the concentration of different antibiotic genes before the treatment, after the treatment, and after 3 days of storage. And in general, you can see that the degradation of the antibiotic-resistant genes were not sufficient, and any of the processes were enough strong to degrade the antibiotic-resistant gene. And even sometimes, in some cases, or in most of the cases, the concentration of these genes are higher after 3 days of storage. But the main conclusion was that the photophantom carried out at neutral pH was the lowest efficient processes in general, considering the degradation of all the targets. Meanwhile, the combination of titanium dioxide P25 with hydrogen peroxide showed the best compromise for inactivation of bacteria and degradation of removal, although it was not able to avoid the regrowth, and the regrowth especially focusing on the antibiotic resistant genes. So summarizing the treatment available today for wastewater treatment, as you see, we have the chlorination, the UVC radiation, ozonation, and also the chemical oxidation. We still have many disadvantages, and we need to still working more for obtaining more effective processes to rate the efficient, not inactivation or degradation of contaminants, because these processes can be done. We must to focus on the degradation of antibiotic resistant genes, because they are the key point and the most resistant target in the wastewater to be removed. And so finally, for concluding, just to highlight that antibiotic resistance of bacteria is a reality and a challenge for the coming years. In the application of efficient processes for the reclamation of wastewater, the elimination of antibiotic-resistant bacteria genes and the organic antibiotics must be considered, as well as the microbial regrowth after treatment, to have a full, complete view of the efficiency of the processes. We cannot forget any of these points to obtain the best conclusion on the efficiency of our processes. In general, conventional water disinfection processes have high efficiency in the control of bacteria and antibiotics, but their effect on genes is relatively weak, generally due to the application of insufficient doses that do not guarantee the safety of the treated effluent. Currently, the efficiency of new sterilization methods, such as the advanced oxidation processes driven by natural solar light, on antibiotic resistance is still relatively low. The findings indicate that the removal of antibiotic resistant G is less effective than the bacteria. Therefore, it's important to find new systems, new photocatalytic materials, new photochemical processes efficient enough to eliminate successfully the genes to avoid its transfer in water. And in this way, probably the combination of sequential processes could be of great interest. Although more studies are needed to optimize parameters and to improve the treatment effect on economic benefits. So that's it. Thank you very much for your attention. And just to tell you that this is a picture of the people and of the solar water treatment plants we do recently with enough separation because of the COVID situation.
Thank you very much, Inmaculada. It was a very nice presentation and very informative. So now I would like to move to the questions. I'm going to go over the questions that were provided within the chat. So the first question is about antibiotic resistant bacteria. The question is that our antibiotic resistant bacteria is spreading along the sun belt. Is our ARB related to high temperature regions, tropical, or it is because of the progress level in terms of wealth, science, etc. of the country?
It's not depending on the weather or the region. It's depending on the use and abuse of the antibiotics, because at the end the humans are using those antibiotics, and then the residual water go to the wastewater treatment plants, and in that point is where it start the problem. So of course we need to use antibiotics, but the problem sometimes is the abuse on the use of those antibiotics.
Which answers our second question, that that it's because of the abuse that humans are using the antibiotic wrongly. And do you know how they actually determined that map? Maybe the methodology also has something to do with it.
Yeah, exactly. All this problematic has been detected and appeared during last year because the scientific community start to know that this is happening and start to increase the studies developed specifically for detection of this antibiotic resistant bacteria and genes in not only in wastewater, also in surface. It can be also detected in surface water, in well water, especially in urban or peri-urban places where the water is not well treated. It can be also sprayed in other supposed clean water bodies. And
so the main idea is to have antibiotic itself present so that bacteria will gain resistance, right?
Exactly,
in the
because
body.
exactly. And also along the not in all places or countries in the world we detect the same type of bacteria, because also depend on the type of antibiotics that in the medicines are using.
And the next question is, do you think chemicals used for wastewater treatment increase the antibiotic resistant bacteria? The other chemicals that are in wastewater treatment?
Other chemicals apart from antibiotics, not, because at the end the resistance of the appearance of bacteria resistant to antibiotics is because they try to survive and adapt to the presence of the antibiotics. So the bacteria generate mutation, and this mutation that are perfectly for avoiding the effect or the attack of the antibiotics is it's where appear the antibiotic resistant bacteria. So other chemicals are not directly related with the appearance of antibiotic resistant bacteria.
And the next question is there any chance that bacteria might also get resistant to the inactivation processes, such as chlorination, ozone, or UV? And if yes, do you think it's possible to have, if it's possible, which of the inactivation technologies would be more reliable compared to the others if they are developing any type of resistance?
Well, as baseline, we have different. When we in the wastewater, we have different groups of microorganisms with different resistance to the processes. This is natural. For example, the bacteria normally are the lowest resistant to treatment in comparison with viruses or in comparison with spores of bacteria, because they are more resistant. But if we speak about the differences between naturally occurring bacteria and antibiotic resistant bacteria, there is no differences on the resistance to this treatment, because they have the same architecture and the same capability to react against the processes. So there is no difference in that sense. You have differences if you are comparing different groups of microorganisms in the water. Like I say, bacteria are lower resistant than viruses, than spores of bacteria or fungi, etc. But not differences between E. coli and resistant E. coli bacteria. Then in the group of bacteria, typically there is a differences on resistance depending on the type of bacteria. The gram-negative bacteria typically are less resistant than the gram-positive one because of the structure of the cell wall. Because, but you mean this is the main differences in the different group of microorganisms.
Mm-hmm.
And the other
so
thing,
I'm moving to the next
thing
no,
is
yeah,
possible.
and the other thing is, yeah, you can see the question too. And if there is a difference, is there possible to have a more reliable technology? Was the question. So, but you already answered it, that they actually are that there is no chance to get resistant to all of them, and the possibility was different based on their structures. So, do you think that one of the technologies are better than the others? Chlorination, ozone, or UV?
For example, chlorination is a process that is used because it's very effective for the inactivation of bacteria, and the cost is very low in comparison with the other technologies. But it has the problem of the generation of the compounds called disinfection byproducts that even more problematic for the environment, and it has more risks. So it can be avoided, the chlorination, but due to the cost, they still use it.
Mm-hmm.
So other processes like UVC or advanced oxidation processes based on UVC or even ozone, but always that you keep some residual concentration of a secondary oxidant. For example, in a combination of UVC with hydrogen peroxide. This is a very powerful treatment, but you must keep some residual concentration of this hydrogen peroxide to avoid the regrowth. And also,
and that's my question. If you keep the secondary residual like in very low concentration, would that also kind of lead into sort of a resistance, or would bacteria get used to this chlorination doses? Maybe that was the baseline of the question, because they might be getting more resistant to the chlorination or to ozonation, and then we have to keep increasing the concentrations gradually, which is something negative.
It can be occur if you apply a treatment with a chemical oxidant, and you don't kill completely this bacteria. After certain times, it appear as regrowth microorganisms. Maybe it's not even the same strain that you have intended to kill, but the resistance even can spread to another environmental cells. So the regrowth is very important, and also it can appear what you say if some sublethal damaged bacteria after the treatment regrowth, they can be sometimes more strong to other potential effects. And you will need more for eliminating. You will need a higher concentration of other processes. So the important is when you apply processes, you must to be sure that you are killing bacteria and degrade the antibiotic resistant genes that, as you know, in all the treatment that we have right now in like conventional and solar advanced oxidation processes, they still being present in the water. And if they're still being processed in the treatment, and you discharge this water to the environment, these genes can be spread to another strain.
So which probably we can tie to the next question about technologies. So can we engineer a biological entity to deactivate the antibiotic resistant gene? I think that's what the next question wants to say. Is it possible to create human animal friendly bacteria to kill the antibiotic resistant bacteria. So basically, can we engineer something? Because you spoke about the enzymes too, so maybe that's how the question is related. And the second part of the question is, are there any research about microalgae that can consume the antibiotic resistant bacteria?
Well, it's interesting that the last question. I don't know if they exist. I know that they exist in the environment. Microorganisms that eat other microorganisms, but even in these processes, the antibiotic resistant genes is still being in the. It's like you eat, but the genes is inside the bacteria. Is the removal, but the genes is inside. Maybe I don't know right now any work related with the microalgae specifically. But it could be interesting to check and see about that.
Especially if the antibiotic resistant, yeah, especially if the antibiotic resistant genes are actually targeted. That would be interesting to know if any type of biological entity could target the gene that also could be horizontally transferred, right? So now the next question is about parabolic collectors. Why not parabolic through or linear Frenzel collectors, but parabolic collectors?
Okay, yes. Well, I think it's got the Thailand know very well the type of concentration system that we have at the PSA. The parabolic throw collector and the linear and the Fresnel collector and other types like these collectors are a system for solar thermal concentration system, for high solar thermal concentration system. However, for water treatment, we don't need this solar thermal concentration system because they are highly cost. And they are very, very highly efficient for generation of electricity, and that's kind of activities. For water treatment, we look for photoreactors as low cost as possible and as efficient as possible. And the collector, the CPC, the compound parabolic collector, was developed at around 20 years ago, more or less, and this is in the medium of flat system that are the lowest cost and the high solar thermal concentration system, like the collector through the parabolic throw collector, and are less cost because they are a static system. They are not trekking because for high solar thermal concentration system, you need the trek for searching the movement of the sun because they need the direct radiation. But for solar application, for the solar treatment, we don't need this effect. With the CPC, we can collect the radiation and increase the efficiency inside the reactors with a more sample and low cost system, and we don't need high pressure in the system. So the compound parabolic collector is like a special development for the application of water purification by solar advanced oxidation processes, and it's less costly than the big system.
Yeah. Thank you for the question, because that was something that I actually had missed from the talk, and I learned too. So the next question is, did you ever try the titanium oxide plus semiconductor photocatalytics, which they claim that might be more effective on water treatment? So basically, a semiconductor photocatalyst with an advanced oxidation technology. In this case, apparently it's titanium oxide.
Yeah, we have working with titanium dioxide, the commercial one, the more efficient catalysis the titanium dioxide to P25. But also we have evaluated and we evaluated continuously new modified semiconductor that permit increasing the absorbance of more wavelengths in the range of the solar radiation. But today, titanium dioxide P25 is still being the most cost-effective semiconductor, and we have evaluated the degradation of antibiotics, the inactivation of bacteria, and the result that I have shown you in the presentation. But we will still evaluating the titanium dioxide with other agents like hydrogen peroxide or other possible oxidant like persulfate and other possibilities to try to increase the efficiency of the semiconductor to be more effective in the wastewater treatment.
Okay, and our last question in the chat is based on why antibiotic-resistant genes are not degraded. It says, did you do the AOP degradation of the ARB and ARG together in the water or separately?
Always are together. So when you in a typical experiment of wastewater treatment, we take the secondary effluent of a wastewater treatment plant, and in that water you have the naturally occurring bacteria, and inside the bacteria there are the genes. So you cannot separate. Well, you can separate if you spike specifically some genes or some bacteria. But when we evaluate the wastewater, everything are inside. Concentration of antibiotics, the bacteria, and also the genes. See,
then the question would be why the antibiotic resistant genes are not degraded when the bacteria is deactivated, I think.
Yeah, we evaluate it all together, and we see that the bacteria is inactivated, but the concentration of genes is still being in the sample because the process has not been enough to reach a total mineralization. Because in an ideal case, the advanced oxidation processes, specifically that where you are generating a lot of hydroxyl radicals in ideal scenario, you will obtain a total degradation of the organic contamination. But normally this is not occur because you need treatment, very long treatment times. But anyway, the answer is we did all together. The bacteria are killing very easily, but the antibiotic resistant genes are still being detected in the water.
Mm-hmm. So if anybody else has any other questions or clarification, you can write to the chat. And I have one question to you before we close this entire presentation. So we spoke about the antibiotic resistant gene and the wastewater. And I've been thinking, like, how does the bacteria actually reach up until the wastewater? And I wanted to hear what your thoughts are to be able to decrease the resistance that occurs throughout those bacteria and pathogens. Do you think treating it in wastewater is a solution, or is there something else that could be done as technology that the antibiotic resistance can be at least minimized globally?
Well, globally is requires first that the population do the things with the use of antibiotics in the proper way. This is the first point. The second is when reaching the wastewater treatment plant, most of the treatment plants has the primary treatment and the secondary treatment. So it is important that that in the treatment plan appear the tertiary treatment, which not occur in all cases, but the application and the implementation of this tertiary treatment must be powerful enough, because we know that this problem exists. That must be powerful enough. And also we need more research and more studies on the not only the inactivation but the treatment, but also in the capability of the spreading of this bacteria in the environment. I mean, what happened in the case you have in the treated water, for example, one log, you detecting only one log of antibiotic of the specific genes. Maybe it's not important because they are diluted or it is important. So I mean, we need more research, and more knowledge in that point. So we know that it's important to reduce as much as possible, but also what happens after that. The impact is higher, is lower, the risk is higher, lower, depending on a lot of variables. The receiver's water body, or if the water is used for example, irrigation in agriculture. What happened with these genes if you don't treat enough the water and then are using for irrigate crops that then coming again to the humans. So still being requires studies in this part of the total scenario.
So basically, the problem is not that antibiotics and bacteria comes from the sewer and travels, but it's actually inefficient treatment that could be in the effluent, and then effluent can be spread all over. So that is why we are focusing on the wastewater treatment plants. I just wanted to make sure to clarify the topic. Thank you so much. I think we answered all the questions, and it seems like everybody is quite happy. We don't have any more comments. So thank you very much for attending today, answering all our questions, and presenting this interesting topic.
Thank you. Thank you, all of you. And thank you for joining the webinar. Just in case you have any more information, just you can search my mail in the presentation, and also in the website of the PSA. You can find more information about all of us, our activity, and you can send me an email if you want more specific information in a topic. So I'm available for anything.